Um, Today, I'm going to talk to you about brain decoding from large quantities of data. To begin with, I want to provide a little bit of motivation and talk about why this is an area that I think is particularly interesting and important in the coming years. So we now have fairly sophisticated technologies for recording brain activity. We have both invasive and non-invasive techniques. The non-invasive techniques are improving in their resolution and portability. We're seeing, for example, optically pulsed MEG helmets that could potentially give us high-resolution, non-invasive, but freely moving recordings. Invasively, we're seeing the number of individual neurons that we can record from increase every year in a sort of Moore's law for neuroscience. And all of this neural activity data that we are receiving is informative about people's behaviors, the stimuli that they're experiencing, and in even their internal mental states, and of course their health states, we should say. So in principle, if we could decode neural activity, that is, if we could interpret a recording of neural activity and from it say, okay, here's what this person was thinking, or here is what this person's health status is, or here is what behavior this person was engaged in, there would be numerous downstream commercial and clinical applications. And we could really see, I think, the emergence of a robust neurotech economy in the global market. And of course, we could help many patients suffering from neurological diseases. The challenge is that neural activity is very complicated. So neural activity is ultimately sampled from this very stochastic, very nonlinear dynamical system that is itself distributed across a large number of neurons, basically rendering it a very high-dimensional, nonlinear stochastic dynamical system. And analyzing such systems is a big challenge. Really, we can't accomplish that with standard statistical techniques. Running linear models, even nonlinear models like kernel models, or any kind of simple hypothesis testing or regression is generally not going to cut it. And really what we need to work with such complicated data, with such high-dimensional, nonlinear stochastic data, is deep learning. I think at this point in time, the vast majority of people are on board with this claim. But if we're going to employ deep learning, we cannot forget the bitter lesson. Now, the bitter lesson refers to an idea first articulated by the AI researcher, Rich Sutton, who wrote a blog post in 2019 titled The Bitter Lesson. And in the blog post, Sutton observed that, generally speaking, the greatest success in AI over the last 20 years, now 25, has come from the application of very general purpose systems that can be applied to large volumes of data and which can take advantage of large amounts of computational infrastructure. So this is important because there might be a temptation to think that what we can do is design very specialized systems to work with small amounts of data, and that will ultimately achieve what we want to achieve with AI systems. But what Sutton argues in his blog post is, look, to date, there's no evidence that we are sufficiently smart to design appropriate specialized systems that can operate off of small amounts of data. Instead, it looks like we have to embrace the complexity that exists within the data and really let the data speak for itself by using techniques that can be applied to large amounts of data in order to extract the patterns that exist within that data. So it was called The Bitter Lesson because perhaps it's a bit bitter to learn that really we're not smart enough to design specialized systems. But I think it's something that has proven itself to be true repeatedly since Rich wrote his blog post in 2019. The greatest advances in AI have come not from specialized systems generally, but from very general purpose systems that can be trained on large volumes of data. Even though there can be ways of doing it efficiently, as we saw with DeepSeek, that's still ultimately the best strategy for deep learning. Now, what I'm going to talk to you about today is the unique challenge that we face with neural data in respect to this question of how can we train on large scale data? And in particular, how do we overcome the diversity that exists in neural data sets? Let me explain a little bit what I mean by that. So we face a challenge when we seek to train across large volumes of neural data. Remember, of course, that neural data is very different from, say, text data on the internet, in that it is collected through relatively painstaking procedures. It is typically collected for different purposes. So it is collected for different types of studies, different clinical questions, etc. And added to this, every brain is unique. So when we do a recording from your brain or my brain, one of the issues that we face is that we don't share all the same neurons. There's not an identical pairing of neurons between individual humans, or even between individual mammals of any sort. So unless you're a C. elegans, you have a unique set of neurons in your brain compared to every other human being. And those neurons are laid out in a unique shape with unique folds in your brain, etc. And as a result, it really is the case that there are no two neural data sets that are exactly alike. Another way of framing it is that every neural data set is in a different language. So, not only are the neurons in each brain unique, the behaviors and the stimuli that are being used in the experiments are distinct. The recording modalities can be different. We have different species. The health status is diverse. And so it's kind of akin to a situation where, let's imagine we were trying to train a large language model on text from the internet, but every website had its own language. This is the challenge that we face with neural data. And it's not immediately obvious how we can train on all of the neural data that we might, in theory, be able to get our hands on, given that it is all so diverse. Put another way, what we need is a sort of universal translator for brains. We need a model that can put every brain's unique language into a shared space, a shared latent space, such that even though my brain's different from your brain and one recording modality is different from another recording modality, etc., that activity can be ingested by an AI model and put into a latent space where they are ultimately merged and their shared grammar, as it were, is identified. So that's what we have been seeking in my lab and with our collaborators at a variety of universities, is how can we design systems for creating, as it were, a universal brain translation machine. Now, one way of framing this problem is how can we tokenize neural data in an appropriate way? So when I use the word tokenize, if you're unfamiliar with natural language processing in AI, let me just explain a little bit what I mean by this. Artificial neural networks that we use in deep learning ultimately ingest vectors. That's what they work with. High-dimensional vectors, so long lists of numbers. In order to get a neural network to process, for example, linguistic stimuli, what you have to do is you have to embed every word into a high-dimensional vector space. This is a process referred to as tokenization. In this example, we have the word dog getting embedded into a high D vector space, and and notice that ideally there's some meaning to the tokenization to some degree, insofar as you might have situations where words that have similar meanings, like dog and cat, are close to each other in the tokenized vector space. Now, in neuroscience, we have a similar issue. We want to somehow tokenize our data. So we want to take activity from different neurons in the brain and turn that activity into a series of high-dimensional vectors, effectively placing the neurons of the brain in a high-dimensional space. And ideally, we would do so in a manner that makes that tokenization meaningful, that is actually informative about how the different neurons relate to one another. Now, the approach that we took to begin with to start to build our universal brain translator is we started with invasive data. I want to emphasize that everything we've built here can be extended to non-invasive techniques, and I can talk later about how we do that. But for now, to make things easier, for sake of explanation, it's worth just being very concrete about the first set of steps we took. So what we initially did is we were working with invasive electrophysiological data, and we decided that we were going to take an approach inspired by natural language processing. In natural language processing, the way that you deal with sequences of words is that you create a token, typically one for each word. Sometimes a pair of words or a few words will get grouped together. Sometimes subwords will be a token, but generally on average, it's going to be roughly one word per token. And we then, in analogy to this, decided to treat each spike like a word from the neurons. So we create a token from every spike that we identify in the recording. And the way that we do this is through a two-step procedure. The first step is that we create a very simple input token that is defined in the simplest possible way. It is essentially a vector that indicates the specific neuron from the specific recording session. So it's literally a number that just says, this is neuron 59 from recording 63. We then add to that a timestamp that says when the spike occurred, just relative to the start of the recording. So this is neuron, whatever, 67 from recording 53. It occurred at time 512 milliseconds. And this information is turned into a D-dimensional vector. And that vector is what we then feed into our neural network. So importantly, in this initial way that we set it up, there is no meaningful information. Every neuron receives an arbitrary unit ID, and the timestamps are also ultimately arbitrary because they're all relative to the start of a recording, and that can be different in every recording. That is where the start is relative to some behavior or stimuli or whatever. So how do we then turn this into a set of meaningful tokens, and how can we operate with the diversity that exists in different neural recordings? Well, what we do is we use individualized embeddings with a perceiver I/O architecture. So I'll explain what I mean by that. First, just by way of explanation, here on the left, what I have is the traditional tokenization procedure, the traditional self-attention procedure, I should say, for transformer models. So what happens is you receive a series of tokens at the input, and remember, each token is going to be roughly a vector representing a word. From those vectors, you produce a series of new vectors, a query, a key, and a value through a matrix multiplication operation. And then you compare the keys and the queries of every token to the other tokens, and you determine which of those keys and queries match best. This gives you what's referred to as your attention weights. The attention weights are then multiplied by the values of each of the tokens, and the new tokens are a weighted sum of the values of the tokens in the previous layer, where the weighting is determined by the match between the key and the query via a softmax function, usually. This procedure is great for allowing tokens to basically learn the relationship between each other. So if I'm a token that is the word dog, for example, I will develop key and query matrices that are on the lookout, as it were, for things like furry or friendly or tail or other words that will typically have relevance to the word dog. And that process will then allow the system to learn about the relationship between all the different words, including how those relationships then relate to the meaning of the sentence. And this is why transformers are very effective at processing language. Now, we face this unique situation that in our case, the tokens are at first all different from one another and arbitrary. They don't have any clear meaning, and it's hard for the system to work out across different data sets where we have different neurons spiking in different ways what the relationship is. So what we do is we use this perceiver IO architecture shown on the right here. Again, we have our tokens fed in. Here, each token is, recall, a token about a spike. And so it indicates which neuron it was and when the spike occurred in the recording. And then we generate keys and values just as we would with normal tokens. But what we do at the first layer of the network is we instead use a set of learned latent tokens to generate the queries. Now, these are vectors that can be learned, and they can be fine-tuned for every data set. Why is that important? Well, because we can fine-tune these quite easily for every data set, they can function as a little lookup table, as it were. They are basically a translation device that says, okay, in this recording in this neuron from this recording, a spike at this time means this relative to the other spikes. Whereas in this neuron from this recording, a spike at this time means this different thing relative to the other neurons. So we've got this ability to treat every token in a different manner and have them relate to each other differently for each data set. This is what gives us the ability to train across diverse data sets. So this perceiver IO architecture, coupled with the simple tokens that we generate for every data set, allow us to train across many, many, many data sets and have the neural network learn itself how to relate these different spikes from different individuals at different times. So in the first iteration of our model, this is what it looked like when we brought it all together. We named the model Poyo because the grad student who built it was a fan of Kirby. So for those of you who play video games, and the idea was the model wants to eat all the data, much as Kirby wants to eat everything. So here you see an illustration of the principle I've already described to you on the left. So we've got spikes from different data sets. The rainbow coloring here indicates different time points. And for each spike, we generate one of these simple input tokens that says, I am a spike from neuron X in recording Y, and here's the time at which I occurred. We run those simple tokens through our cross-attention mechanism. And for every data set, we can then learn that lookup table, that set of learned latents that I just described to you, where we've got a different latent for different time points in the recording and dimensionality of 32. So these are a set of 32 dimensional vectors for different time steps in the recording that basically say, here's how you should actually attend to these different spikes in different recordings. So we mix it from different recordings. Here we've got data set 1, here we've got data set 2. All of the tokens are fed into the same model in the same sequence processing. And in that way, the model can learn the relationship between different data sets. After generating our initial token Z1 through this cross-attention, this perceiver IO cross-attention mechanism, we then do L-1 layers of self-attention, that is regular transformer processing. And then we run that through one last layer of cross-attention. Now, the reason we do that is in this last layer of cross-attention, we generate another set of tokens that are used to indicate what we want to decode from the model. So we provide, again, a timestamp for each of those decoding items, each of those decoding queries. And here we now provide different vectors indicating what sorts of things we want to decode. So, for example, it could be the behavior of a person or an animal during the recording, or it could be what stimuli they're currently looking at, or it could be their current mental state or their health state. These tokens are highly flexible. We can basically just define them as we want, and we will give these different tokens to the network as queries to this cross-attention mechanism. So in this last step, the system is able to look across all of the tokens that are at this point in time a mix of all of the spikes across all of the different recording sessions. And it can say, okay, in recording session 2, we want to decode right now the position of the eyes, say, of the person during the recording. And so that will then be decoded by an output layer. Now, in the first instance, what I'm going to show you is data from some monkey experiments where the monkeys were engaged in reaching behaviors. And so what we decoded was the velocity in horizontal and vertical directions, X and Y, of their hand on the screen, or the joystick, the cursor on the screen, basically, that they were controlling. But I want to emphasize, and you'll see this towards the end of the talk, that this can be applied to any output. So it doesn't have to be hand velocity. That's just what we did in this first thing. Really, this is a very general model that just says predict some behavior or stimuli or mental thing at this timestamp under the experimental conditions of this session ID. So here's the initial data sets we applied this to. We collected a set of data sets, from six different studies in non-human primates. Again, we wanted to start with not too much diversity. Ultimately, our goal is to get up to the point where we can train across different species, and I'll show you some of that in a second, and across very different tasks and brain regions, but let's keep it simple. Let's just start with addressing the problem of diversity of subjects and a little bit in terms of the behaviors. So we've got 6 different studies from primates here. Most of the recordings were done in primary motor cortex, but we also had some in premotor and somatosensory cortex. There were a total of 11 individual monkeys, and we had a couple of hundred sessions, essentially just under 200 sessions that we were processing. You can see here the total number of neurons that we were processing, so on the order of 25,000. And then here, this is actually a list of the number of tokens for each study. So in this case, this is the number of input tokens, which basically corresponds to the number of spikes. So we were processing about 1 billion spikes from across these 6 data sets, and we were outputting about 100 million tokens of decoded information. Now, there were 4 different tasks across these 6 data sets. There were a center out reach with a joystick, getting a random target with a joystick, getting a random target with a touch screen, and getting a cursor to be in a particular location in a maze just by controlling a touch screen. Now, as I said, our goal in this first instance is to decode the velocity of the cursor from the neural recording. Here you can see some example traces of that. So this is from 3 different monkeys, monkey C, monkey M, and monkey JA. And in black is the actual ground truth of the cursor velocity. And in green, we see the predicted velocity from our polio model. As you can see, it's doing a pretty good job of decoding. We can measure that directly with an R-squared value. Here you can see the R-squared values. Now, what I'm plotting on the left here is the R-squareds we get if we train on a single monkey session versus if we train on the 3 monkeys sessions. And this is important because when you train across multiple sessions for multiple individuals with machine learning techniques and neuroscience, often you don't get any benefit. And that's because of that diversity in recordings that I was mentioning. It's not necessarily the case that when applying fairly simple procedures such as decoding from the rate of fire, that you're going to get meaningful signals that will allow you to actually improve your accuracy by training across multiple individuals. But what we see is that when we train this model on 3 monkeys rather than 1 monkey, we get a boost in the R-squared of about just under 2%, which isn't huge, but it's importantly in the right direction. And it indicates that if we increase the size of the training data, we could continue to improve the accuracy there. Here we see across all of the sessions, the relative plot of the single session performance versus the multi-session performance. And you can see that the R-squared is always higher in the multi-session case, indicating that this model is indeed able to take advantage of more data. This is a set of plots further emphasizing that point. So here we are plotting the average R-squared that we achieve in the center out task as a function of the number of parameters of the model that we train. So with a relatively small model here with just 3.8 million parameters, if we move from a single session to multiple sessions, we get this boost that I've already showed you. And then if we continue to increase the size of the model, we can continue to increase the size of that, the value of that R-squared. We see a similar thing with the random target. Single session is worse than multi-session. As we increase the size of the model, we increase performance. So in principle, we could continue to increase the size of the model and the size of the data set and continue to push up that R-squared. Here you can see that plotted a slightly different way. For the random target task, we're plotting the number of tokens that were processed by the system. So when we process a single session, we're processing about 1.2 million tokens. As we push that up to 26 million tokens by including all of the random target tasks for these monkeys, we can increase our R-squared. We can increase it further by adding in the center out tasks, which brings us to 134 million tokens. And if we increase the size of the model from 6 layers to 24 layers, we can increase that further. So in principle, we should be able to scale up the amount of data and the amount of the size of the model in iterative procedures and eventually get to very performant models that are trained across large swaths of data. Now, lest you ask, well, isn't this deeply inefficient? Well, you would be right that the pre-training itself can be deeply inefficient. But importantly, once you've pre-trained a model like this, you can then engage in fairly efficient fine-tuning of your models. So this plot is illustrating that point, these 2 plots, I should say. Here we're looking at sample and compute efficiency, so sample efficiency on the left. We're plotting for a given number of trials that we fine-tune the model on for a new held-out data set, what is the R-squared that we receive? So if we were to train a simple recurrent neural network on this number of trials, we get relatively poor R-squared until we get up to hundreds of trials. If we have a single session model, our system is slightly more sophisticated, so it can get better results. But if we've pre-trained the model, we can get better results at lower numbers of samples. So we can train on, say, 16 sample trials and get basically to the same level of roughly 90% R-squared on these tasks. Perhaps even more striking is the difference in compute efficiency. So if we train a model from scratch, to get to that level of accuracy, it takes us thousands of training steps. But if we have a pre-trained model, we can then fine-tune the model in just a few hundred training steps to get to the same level of accuracy. So you could imagine using this as, for example, a calibration procedure. Let's say you have a patient or a consumer who has received some neurotechnology recording device, and the model has been pre-trained on other data, you could then fine-tune for that person to calibrate the device with relatively few data points. So maybe cutting hours of calibration time down to tens of minutes. And importantly, the pre-training also seems to work for transfer between species. So here I'm showing you some results where we took that model that we had pre-trained across a bunch of different monkeys, and we then transferred it to a human data set. In this case, this was a data set from Willett et al. There was a patient who had received a Utah array implant in their motor cortex, and they were asked to imagine writing letters, and the task was to decode what letters they were writing. And you can see that if we directly train a model on this task, it gets so-so accuracy in the teens in terms of the error rate for held out letter decoding. Whereas if we first pre-train the model on a lot of monkey data, and then we fine-tune it on the human data, we can actually achieve a much lower error rate. And that's with the same amount of data that this was trained on. So it's really advantageous to pre-train on the monkey data. Okay, now can this work with more diversity? So we wanted to then examine whether we could use this approach in a data set where we not only had different individuals, but where the data was also taken from potentially very different cell types, and where we were decoding a few different types of stimuli. For this, and I should say where we had different brain regions as well that were slightly more diverse than just motor cortex. So here we used the Allen Brain Observatory dataset to test our ability to decode behavior and stimuli. The Allen Observatory is a calcium imaging dataset collected from mice in their visual cortex. They did recordings from 6 different visual cortical regions, including primary visual cortex and a series of higher order visual cortices. They presented the animals with 6 different stimuli, including drifting gratings, noise, natural movies, etc. And they did the recordings in different Cree lines of mice. So these were transgenic animals where the calcium indicator was only expressed in particular types of neurons. So for example, different types of interneurons, here parvalbumin positive or somatostatin positive interneurons, or different types of excitatory neurons, here RBP4 neurons tend to be pyramidal neurons in layer 5, for example. And you can see we had different neurons of these different types from the various different brain regions. So 13 different types of neurons across 6 different types of brain regions. And the question is, can we train a model across all of this data and get benefits from multiple data sets? Now, we had to alter the model slightly relative to the last one I showed you because we're processing calcium traces now. Principle is very similar though. We just create a token for every time step in the calcium trace. The only difference with the spike tokens is that we add an additional value to the token saying this was the magnitude of the calcium trace at this time point. We then process those tokens with our POYO encoder. But now what we call POYO Plus, we add in this multimodal decoder where we can have very different queries asking for different types of outputs. So we can ask either to decode the sensory stimuli, for example, the drift and grading orientation or its temporal frequency, or behaviors of the animal. So for example, its running speed or where it's looking. And we can decode all of these things from the same model using this flexible tokenization procedure. Here are the results purely in terms of accuracy. So you'll see that the accuracies are not as high as we get for decoding motor commands from monkey cortex. We think that that's actually probably because of relatively less data, even though there's a lot of data here. In each animal, we have shorter recording sessions. Moreover, mouse visual cortex doesn't actually like a lot of the static stimuli so much. And unfortunately, Alan only played one movie each time for each animal. But nonetheless, what we can see, and this is the important thing, is that in purple, where we have our Polio Plus model that's trained across decoding multiple different types of outputs, and which is trained across multiple different cell types and brain regions, we consistently get better accuracy in the decoding task than a model trained on an individual task from a specific brain region or set of cell types. Now, what's more interesting about this result, though, is what we see in terms of those latent lookup tables, because it tells us something about how the model is dealing with diversity. And that is that the model is actually learning something about the different cell types and brain regions, even though we never tell it anything explicitly about that. So what we did is we took that set of lookup tables for our model, and we asked, can we train a linear decoder to decode first what brain region the recording was from the latents? And what we can see is that actually we can, with reasonably high accuracy, decode from our latents exactly which brain region the recordings were taken from. Here you can see the confusion matrix for that decoding. So there are some brain regions that are very easy to identify. Apparently, primary visual cortex stands out, whereas, for example, Vis-AM and Vis-PM can be relatively easily confused. But this in and of itself is interesting, and it's reflected on the plot on the right here, because what this reflects, this confusion, is actually some real components of the anatomy. So here we're doing hierarchical clustering of those latent vectors, and you can see that what has happened is that the model has learned to group lateral and medial regions together. That is, lateral regions are together and medial regions are together, and they're separate from each other, which means that even though we didn't tell it anything about the anatomy of the brain, the system was able to learn that indeed tokens from PM and AM are more similar to each other than tokens from AL and SL, which is pretty remarkable. It suggests that there are some underlying traces of the dynamics in each brain region that are informative and unique to those brain regions and which reflect the underlying anatomy of the brain. We see a similar thing with the cell types. So here we're decoding, actually I'm not decoding yet. This on the left is a t-SNE plot of the latent vectors for the recordings, color-coded by whether the recordings were from an excitatory or an inhibitory cell. And what you can see is that there is clear clustering of the inhibitory cells together as distinct from the excitatory cells, or at least many of the excitatory cells. You can see this reflected in the confusion matrix on the left here. So again, this is just the accuracy of the linear decoder across the different classes. So the inhibitory neurons, it's quite good at classifying and doesn't confuse too much, neither with the excitatory neurons nor with each other. It has a little bit more trouble across the different excitatory types, but that makes sense because some of these are overlapping. And yet, nonetheless, it's actually able to identify them with a fair bit of accuracy and certainly well above chance. Moreover, per what I showed you about the anatomy of the brain regions, it actually learns something about the distinct physiological properties of the cells. So, for example, one of the things that characterizes these different Cre lines is that they target different types of neurons with different types of projections. So, for example, there are these are intratelencephalic neurons, that is, neurons that project within the telencephalon. We have then pyramidal tract neurons that project down to subcortical structures like the brainstem, and cortical tract pyramidal neurons like cortical thalamic pyramidal neurons that project down to the thalamus, for example. And when we run our hierarchical clustering on those latents, again, the inhibitory cells come out as distinct from the excitatory cells, as was expected. But moreover, what we see is that the different types of projection neurons will largely cluster together. So the cortical thalamic neurons tend to cluster together. The pyramidal tract neurons tend to cluster together. And that's really interesting because it says that even though we haven't told the model anything about the different projections of these different neurons, it's able purely through the dynamics of the spiking to identify different classes of neuron based upon their spiking profile and relate those to each other in a manner that reflects the actual projection anatomy of the neurons. So, to wrap up, neural diversity is not something that need be feared for neural decoding. We can scale things up, and our results, both the results I've shown you today and some additional research we've been doing over the last few months, indicate that indeed neural data can also follow scaling laws. You can increase the scale of data on which you train and achieve better accuracy in your neural decoding, as you do in other domains of AI research, but it requires these special techniques to attend to the diversity that exists in neural data sets. So that sounds very good, but let's just run a little back of the envelope calculation here. If we really want to take advantage of scale, what would we need to get to in terms of the data set size? Well, as a rough estimate, let's say that we want to train our models on roughly the same scale of data that we train large language models on. So those are typically trained on trillions of tokens. Sometimes the larger ones today are trained on tens or hundreds of trillions of tokens, but let's just stick to trillions as a order of magnitude for a moment. So let's imagine that we're taking neural recordings that have roughly 100 neurons in every recording, with each neuron spiking at roughly 10 hertz on average. That'll give us 1,000 tokens out of every second of a neural recording, which means that we'll get 3.6 million tokens in an hour-long recording. If we want to get to over 1 trillion tokens with this level of number of spikes in a given recording, then we're going to need roughly 300,000 hours of recordings. That sounds like a lot. It is a lot, but I think it's notable that it's within the realm of possibility, given the number of recordings that are done every year across the entire globe. In principle, we should be able to get to hundreds of thousands of hours of neural recordings and train a model across those. So ultimately, this is one of my personal goals over the next 10 years, is to train models at this scale on neural data. And that's because we have these sophisticated technologies now for peering into our brains. And I think that if we develop equally sophisticated technologies for decoding neural data, it will really open up a series of commercial and clinical applications that don't currently exist. And for that reason, I'm very excited about this potential and want to pursue this at scale. That's that. Thank you very much for listening. Sorry again for having been late. I want to thank my collaborators on this, most notably Eva Dyer and Mehdi Azabu, who have worked very closely on this over the last few years, as well as my good friend Guillaume Lajoie and my colleagues Joel, Matt, Patrick, and Crystal. We had a paper at NeurIPS on our first model in 2023 that you can see, and we have the model for the Allen data coming out at iClear as a spotlight paper this year. Thanks very much for your time.